Thank you, Elizabeth, and thanks also to CHI and Personal Genome Diagnostics, and especially Melody Gretz and Melissa Reuter for organizing this webinar. Looking forward to this webinar together with Drew and with Teresa. It's going to be a three-part webinar, as you've heard, and the first part is focused on really understanding the genomic characteristics of cancer and how these lead to neoantigens and what their utility may be for immunotherapy. Now, we all know cancer is a genetic disease. This, of course, came about through work over the past several decades identifying individual cancer genes, but was really highlighted by work that began about a decade ago, expanding those analyses to entire cancer exomes and genomes. And this is an example of the first 100 cancer genomes that were done initially through Sanger sequencing up to medulloblastoma, starting with colorectal, breast, pancreatic, and GBM, and then extending using next-generation sequencing approaches to pancreatic neuroendocrine tumors, ovarian clear cell carcinoma, head and neck cancers. And these were studies that were largely spearheaded at Hopkins, but were very nicely extended through the TCGA effort and through the ICGC by many other institutions worldwide. These analyses really defined for the first time the genomic landscape in human cancer, identified a number of new genes and pathways that are important in human cancer, some of which are listed below, and open up new avenues for therapeutic and diagnostic intervention. One of the ways in which these types of analyses have, of course, been useful is through the understanding that there may be new targets of therapeutic intervention and may also identify actionable changes in individual patients. This was nicely shown in a study by Sean Jones and colleagues at both Personal Genome Diagnostics and Johns Hopkins that looked at over 800 tumor normal samples from patients of a variety of different tumor types using both whole exome as well as targeted analyses. This study was hopeful in the sense that it showed that the majority of patients could potentially benefit from having this kind of genomic information. As you can see in this graph, a large fraction of different tumor types have actionable alterations when you analyze their cancer genomes or target analyses. But the caveat to this, of course, is that very few of these are FDA-approved therapies, as shown in the dark green, or therapies that are known to dramatically work in these patients. Many of the genes that are actionable right now in cancer are those that are in clinical trials, which are shown here in blue, and time will tell whether these are actually helpful for patients. Now, the second way in which this kind of information can be helpful for individuals is not through small molecules or biologics, but potentially through immunotherapy. And this is something that many folks have been working on really for a very long time, and Drew is a great example of that, and he'll delve into it in the second part of the presentation today. But there were already links between genetics and immunotherapy some time ago. This is an example that we can see here from work done collaboratively with Jim Allison back in 2008, looking at the very first cancer exome analyses in colorectal cancer and in breast cancer, showing that if one looks at the theoretical neoantigens that come out of these studies, all the possible mutations, and asks the question of whether any of them are likely to be presented in an HLA-restricted fashion, you see that at least one mutation in each case is a potential neoantigen in every one of these tumors, nicely showing that there is the possibility of recognition by the immune system. And it took a long time for that to mature, and we'll get to that a little bit later in these presentations.